to chemical principles. Most, if not all, of this chapter should be review for you, as you've already taken chemistry and introductory level biology, as well as anatomy. So feel free to put this on, a faster speed just to get through it faster. But I do want to warn you, even though most of it seems very easy or repetitive for you, I find that students still tend to miss these questions on the exams. Maybe they skipped over the chapter to spend more time on the other chapters. So don't miss out on these points. And let us know if you have any questions about this material as well. Okay, so these elements, as you are probably familiar with, are the most abundant in the biological molecules. So in your DNA, in your proteins, and all the other molecules in your body, you're going to find a lot of carbon, hydrogen, nitrogen, oxygen, right? We need large amounts of all of these nutrients so that we can make all of our biological molecules. So you can continue reading down the list of these elements that are the most, these are the elements we need the most of. We find them in large numbers throughout our body. Furthermore, you'll see other elements that serve as ions or electrolytes in the body, and are constantly being put in or out of a cell to create things like action potentials, stuff like that. So it's not just to be integrated into the biological molecule, but sometimes being used for other processes in the body as well. Okay. So organic compounds always have carbon in them. Carbon is the backbone, and everything else gets added onto it. Okay, what else? Yeah. So another thing to remind you about is that you can have the same chemical formula for sugar, C6H12O6, but have different three-dimensional structures, as we see here. Glucose compared to galactose, this is swapped around, and then on fructose we have, instead of having this double bond here, it's here, and then that gets you an extra hydrogen over here, right? So this is a different arrangement, but it still adds up overall to the same amount of carbon, hydrogen, oxygen. So glucose, galactose, and fructose have the same chemical formula, but these structural, what we call isomers, they are actually different in their physical and chemical properties. You need a different enzyme for each of these sugars. And as you'll see throughout our course, some bacteria will have the enzyme to break down additional molecules, and some will not. So some bacteria can only use glucose, and that's it. They're very simple. Others have all sorts of enzymes to do all sorts of sugars. So again, see how there are slight differences between the different types of sugars, and remember that those slight differences in function in the body actually is a huge difference. Because like I said, you'll need a different set of enzymes to be able to build up or break apart these specific molecules, okay? And that's a key concept when we look at trying to identify an unknown bacteria, doing different types of tests in the laboratory, what we call biochemical tests. All right. So how are atoms? What are atoms? How do we know about different types of bonds? So make sure that you can differentiate between an ionic bond and a covalent bond and a hydrogen bond, okay? So ionic bonds are attractions between ions of opposite charge, right? So you have, here we have a sodium ion, and it's positively charged. And then here we have a chloride ion, and it's negatively charged. And so what does that mean? It means that these opposite charges are going to attract so that they can share this electron. So now you can see they will get close together and kind of share that negative charge, so that now they're in balance. And when you put sodium and chloride ions together, that forms a table salt. So when it's dry, it's a salt. When you put it into solution, it's going to go back into being in the ions, separate ions. It's going to dissociate, right? Okay. So the idea is that one atom loses an electron, then the other one gains an electron. Ions are thus charged atoms, and electrons are responsible for bonding atoms together, right? So you can see this electron here in red is what's making the sodium and chloride bond together. They're sharing that. Okay. All right. Covalent bonds. Two atoms share one or more pairs of electrons. The electrons will orbit each nucleus. So here, this is not going around here and then back around here. It's just simply, as it says, one atom will lose the electron, the other one will gain it, okay? For covalent bonds, two atoms share one or more pairs of electrons, where it orbits one and then it orbits the other. But sometimes it will spend more time orbiting one than the other. So you can still get slight charges like we see with water. So one atom might have the pair more of the time and cause partial charges to form, but it's still covalent bond. The electrons are orbiting both nuclei. Covalent bonds are much stronger than ionic bonds. It will take energy to break the covalent bond more than ionic. Okay, hydrogen bonds, on the other hand, are just weak attractions. Right? They're attractions. They're not true bonds. Okay, so hydrogen bonds, hydrogen atom that is covalently bonded to oxygen or nitrogen. And here we see the red is the oxygen and the hydrogen are these gray pluses, right? So within this water molecule, you have a covalent bond between the red and the gray, which is not shown here. They're just they just look glued together in this example. So those are the covalent bonds within the water molecule. But when you have two water molecules next to each other, you get this attraction between the slight negative charge here and the slight positive charge here. And so the dotted line represents the hydrogen bond. Why is there a slight negative charge on oxygen? It hogs the electrons. The shared electrons spend more time with the oxygen than it does with the hydrogen. And so that's what leads to these partial charges on the water molecule. And then that's why you're able to get this attraction between the opposite charges of each water molecule. And so you'll see lots of hydrogen bonds form. And so even though one hydrogen bond is very weak, when you have hundreds and hundreds and hundreds of hydrogen bonds, then it becomes very strong. It will take a lot of heat or a lot of energy to break those hydrogen bonds because there's so many of them. Okay. So we're seeing the example being oxygen here, but we can see the same kind of dynamic when you see a nitrogen molecule with nitrogen in it. And like I said, there is strength in numbers. When you have a lot of hydrogen bonds, that you then have a lot of strength. And in fact, that's what holds our two strands of DNA together into the double helix, is all hydrogen bonds. Within each individual strand, you have covalent bonds. But between the two strands, what's holding those two strands close together to form the double helix are hydrogen bonds. Okay, so chemical reactions will mean that you're breaking or you're making a bond. Okay, And every single reaction is going to involve energy. And you'll see a lot of times, molecule A, molecule B, and then it forms that you put them together, you get a new molecule. So when atoms, ions, or molecules combine to form a new larger molecule, we call this a synthesis reaction. Sometimes the synthesis, you'll see an enzyme with less energy required when you have an enzyme to put them together, and sometimes it's just no enzyme. Okay, so you have to always pay attention to what's going on with the arrow. So we don't put the enzyme in the equation down here, we put it over the arrow. I don't know why, that's just how we do it. And so these types of reactions are called anabolic because you're synthesizing and you're going to require energy to synthesize a new molecule. Again, this is another way to test and differentiate between different types of bacteria, because some of them will have the enzyme and some of them won't. Some of them will be able to make that new molecule and some will not. And then you can do that same concept in reverse for a decompensation reaction to see, can it break apart? If I give it this molecule, can it break it apart? And then that'd be testing for a different enzyme. And we call breaking apart catabolism. And you are familiar with this when you eat, right? To turn all of your food into energy and building blocks, you're breaking down the food, and that's a catabolism. And this will make a lot of the biochemical tests are going to be, if we feed it this type of sugar or this kind of protein, or this, whatever special compound, can it utilize it? All right. Sometimes you're not simply putting together two things to make one new thing. Sometimes you're doing a rearrangement. So exchange reactions, partial synthesis, partial breaking apart, to reconfigure or make different versions of what you started with. So this is sodium hydroxide and hydrochloric acid. So this is a base and this is an acid. When you put them together, you're going to get a salt and a water. So every time you put a base and an acid together, you get a salt and a water. In this case, the salt is table salt, but it would depend on what acid and base you're using, of course. A lot of reactions, especially in biology, are reversible. So depending on how much substance you have on one side or the other, it will push more of one direction. So each direction may need special conditions as well. You might need more heat for it to go backwards, or maybe you need more water for it to go forwards, right? So whatever the condition is, sometimes it's just how much of the reactants you have, other times it's particular conditions, but these double arrows show that it is reversible. Okay, and you definitely would want to make special notes if we ever show you a reaction that is not reversible, because that would be a special trait, right? Because most of the reactions in the body are reversible, which makes sense. Okay, so other conditions could be pH, in addition to temperature changes or concentration of reactants, right? So now we're going to talk about inorganic compounds briefly. Water is the most important molecule, right? All life requires water. It is inorganic because there's no carbon, and it's polar because you have slight charges, right? Slight negative and slight positive. In nature, we can see water exist in all three phases: a solid as ice, liquid, right, water, and gas, vapor, water vapor in the air. And so this is incredible because it is, I believe, the only substance that exists in all three phases naturally on Earth. It's also fascinating because when it becomes frozen, it will actually be less dense and float, right? When you put ice in your cup of water, your ice floats to the top. And this is super important because think about a lake. When a lake freezes over, the ice stays on top and all the life in the water can still go on underneath. If you didn't have this property, then the ice would sink and it would squash and kill everything below it. What else? So we tend to classify compounds in chemistry and biology, so biochemistry, as being either water soluble or not. If it's not water soluble, we can say it's insoluble, or we could say that it is what it is soluble in would be like maybe it's oil, right? It's not, maybe it's not soluble in water, maybe it's soluble in oil. So looking at whether or not it's able to be dissolved in water is a huge property. You'll see that recurring. Another property that you know about, just if you've ever tried to cook anything like boiling pasta, for example, you know it takes a long time to boil a pot of water. If you were to have that same volume of liquid, let's say milk, you would know that the milk would start to boil and even burn long before your water boils. And again, that's because of the massive amount of hydrogen bonds in the water. It takes so much more heat to break all those bonds so that the water can change from liquid to gas. Okay. Acids, bases, and pH. You have a lab devoted to this, which will help you to resharpen your skills. But briefly, let's go ahead and talk about it. So the amount of hydrogen that is in a solution, the amount of free hydrogen in a solution is expressed as pH. Notice P is lowercase, H is capitalized. The equation for pH, we can use the negative log of the concentration of hydrogen. And that's important. You'll be using that equation in lab and you'll need to use it on your exam as well. So remember that as you increase the concentration of hydrogen, your pH number will decrease because you have this negative here. So as the concentration of hydrogen increases, the number pH decreases. So that's why pH 1 is super strong acid. It has a very high concentration of hydrogen. So increasing the concentration of hydrogen increases the acidity and gives you a lower number pH. Okay. Likewise, increasing the amount of hydroxide ions is going to increase alkalinity. Another way to think of this would be to decrease the amount of hydrogen. So hydroxide will readily bind free hydrogen in solution, reducing your concentration of hydrogen and increasing your pH. Okay, most organisms prefer a pH of around neutral. So we say a range between 6.5 and 8.5. Of course, some organisms would not be able to handle any acidity at all, and they need to be 7.4. For example, it can be very precise that they can have a very small range for that particular organism. But if you were to just look at all organisms, you will see the majority of organisms overall will fall somewhere along the pH of 6.5 and 8.5. Okay. Something else while we're talking about pH is to think about the body. We have different compartments of our body that maintain particular pHs. So our eyes will be anywhere from neutral 7 up to 7.3, whereas our stomach is super acidic, 1.5 to 2, right? Skin is slightly acidic, 5.5 but that can fluctuate depending on the specific area of your skin. Your intestinal tract will be slightly alkaline, right? And blood is strictly regulated between 7.35 to 7.45. Okay, very important that we maintain our blood pH in that specific range, which when you take physiology, you'll learn more about why that is the case. So having these different compartments of our body with their specified pH means that you're likely to find certain organisms can only infect certain areas of the body, because some bacteria will not be able to tolerate, in fact, most bacteria will not be able to tolerate the extreme acidity of your stomach, right? But they're just fine in the intestine or on the skin, right? You always, when you think about infection or you're thinking about the relationship between bacteria and people, you want to think about, well, what is the environment that this bacterial colony can even handle, right? Bacteria have their own enzymes that are going to be susceptible to being denatured if the pH is too acidic or too alkaline. Some bacteria have a wide range, and you'll see that anywhere they get access to, they'll be able to infect. So those are organisms that we pay maybe more attention to, we're more concerned about. Okay, so we'll, as the class goes on, we'll get specific examples. You'll memorize specifics about each type of organism. But for now, I just want you to get the concept in your mind, so you'll be ready to utilize that information when we give it to you. Okay, so I already mentioned this, but here's a visual. So acid will dissociate, and you'll have a lot of free hydrogen in solution, and so we call that an acid because you have a high concentration of hydrogen. A base is going to take up free hydrogen, and you will also have some free hydroxide ions, and so that's a base. So the tricky part about a base is that you could write it this way, where you're going to have some free hydroxide ions, but you could also write low concentration of free hydrogen. So these brackets mean concentration, and here you could put high concentration of hydrogen ions in solution. So a high concentration of hydrogen in solution gives you an acid. A low concentration of free hydrogens will give you a base. And then a salt is neither, right? It's neither. We don't have any free hydrogen to deal with. We don't have any hydroxide to deal with. So it is neither an acid nor a base. It's a salt, and you're getting ions. Okay. All right. And then this gives you an example of items you're familiar with and where they lie on the pH scale, right? So I already mentioned stomach acid can be anywhere from like 1 to 2. Okay. Lemon juice is pretty acidic. That's why it tastes so sour, and that's about a 2. Grapefruit's a little less sour. That's about a 3. Wine can vary, but it tends to be slightly acidic, around a 4. Tomato juice as well. And then we have urine and milk around 6. And then we have pure water. Perfect pure water would be a 7. Most of the water that we drink has some electrolytes in it, which is actually what makes it taste good to us, and we need those electrolytes. And so most of the water we drink will be higher than 7. And of course, there's been this trend for what they call alkaline water, around adding so many electrolytes that you get it to about pH 9. And that's fine. It tastes good. Water that's 7.5 pH and water that's 9 pH, they're going to do the same thing for you. There's no benefit biologically to drinking alkaline water. If anything, it makes your stomach have to work harder because you're going to neutralize a little bit of your stomach acid, and then you'll have to make more stomach acid. Our body strictly regulates each compartment of our body, so we do not need to interfere typically unless told so by a doctor for some specific reason. Okay, ammonia, a lot of household products don't really have ammonia anymore these days because it smells so bad and it's actually pretty dangerous. But you should always read your chemicals and know what you're working with. Ammonia is somewhere between 11 and 12, so it's very alkaline. Bleach is even more alkaline. Bleach is dangerous too, even though we are pretty comfortable with bleach because it seems, it's normal, it's common. But you should always make sure that you have gloves on, proper ventilation, and you're using it at the correct concentration. Never get it on your skin and never breathe it in, right? It is very dangerous for us. Oven cleaner will be even more alkaline, and those solutions that you put down your drain to unclog it, like Drano, will be like this lime water. Super, super basic. So even more dangerous than bleach. Always read labels and follow the instructions. Okay, so can you tell me the difference between an organic molecule and an inorganic? And now we're going to look at some functional groups. And I apologize for this being so sloppy, but all I have is a mouse to work with here, so it's going to be sloppy. All right. So remember I said organic compounds always contain carbon. Inorganic compounds are going to lack carbon. If they do have carbon, it's not serving as a backbone like we see with organic. So with organic molecules, a lot of times you'll see, you'll see a lot of carbons linked like this, and then you'll see other molecules or even functional groups added on to the carbon. So carbon is the backbone. Okay, but inorganic, if you do find a carbon, it's probably in a functional group, not in a chain, not a backbone. Okay, and most of the time you just won't have carbon at all. We also talked about the difference between something that can be dissolved in water and something that cannot be dissolved in water. Okay, so that's another important feature or characteristic. Okay, functional groups. So what are these functional groups? You may already be familiar with them from other classes. Make sure you memorize the most important functional groups. Functional groups are what give, for example, all of your amino acids start out exactly the same, and then we put on different functional groups, and that's how you get the difference between the different amino acids, and that's what gives them their behavior, how you get different folding patterns, how you get different interactions and functions within the body. Okay, so a common functional group, OH group, that's called the hydroxyl group. So if you have one carbon with one hydroxyl group, it's methanol. If you have two carbons and a hydroxyl group, it's ethanol. If it's pure ethanol, then it's what we drink as alcohol. But a lot of times in lab, we'll use ethanol, but it has a bunch of contaminants in it, so it would not be safe to drink. So never, ever, ever drink a lab grade ethanol. It can make you go blind or even kill you. All right, isopropanol is what you know as rubbing alcohol, and it has 3 carbons, and the functional group is in the middle. Okay, and we have a lot more. You can keep going with more and more and more carbons, but these are the most common ones you'll see in biology. Okay, So I was telling you all amino acids start out with the same basic structure. See where this R group is? This is where the functional groups get added on to give you the different types of amino acids. But every single amino acid is going to be carbon, hydrogen, and an amine group, NH2. Amine group, therefore it's called an amino acid. Okay, and you know that we link together lots of amino acids to form proteins, and we'll be looking at a little bit more about that later on. A methyl group is this whole orange box here. It's simply a carbon with 3 hydrogen on it. A ketone, which you'll see especially when we're breaking down things like proteins. A lot of times we can get these waste products called ketones. You've probably heard of the keto diet. That's kind of starving your body, making it break down more protein than it normally would, and that will increase the amount of ketones in circulation. This can be dangerous. If you have too many ketones, it can make your blood too acidic, and that could be very harmful for you. Not to mention you will feel awful. So I'll say it now, and you'll probably hear me say it again throughout this class. Balance is key. Letting your body function the way it's made to function is key. Extremes are bad. So don't do fad diets. Focus on having a healthy balance. Okay, next. So the ketone functional group is the C double bonded O, right? And then if you were to have a C double bond O and an H, then you have an aldehyde, okay? And as we already saw with the alcohols, it's any amounts of carbons with an OH group, okay? And we will see this a lot. These are very common functional groups in biology, okay? A few more that you need to know. You'll see the carboxyl group come up, especially in metabolism. The sulfur, we really only talk about how it'll lead to very strong bonds. So it does come up when we talk about protein structure, but that's it. So, you don't really need to worry about that. I don't think I ever really talked to you about ester or ether, sorry. So I'm not too concerned about these. But carboxyl groups come up a lot, and phosphate groups, of course, are super common to give us ATP, cellular energy, and also to give us DNA. So definitely should be familiar and comfortable with phosphate groups. Okay. So whenever I give you these learning outcomes, make sure that you read them and you can answer them. Okay, so what is a polymer? It's a chain, repeating units. So a chain of repeating units. And those units are called monomers. And some monomers are going to be joined together. So how are you going to build that chain? You're going to join them together through a dehydration synthesis, right? Dehydration, right, to remove water. So sometimes instead of saying dehydration synthesis, sometimes we just call it condensation reaction because you're left with water at the end of it. So you're removing water and you're forming a covalent bond between monomers. Those monomers can be identical or they can be different. It doesn't always have to be the exact same monomer over and over and over again. It could be monomer A, B, C, right? In any order, just repeating over and over. So you're just linking a bunch of monomers together, and that gives you, right, poly, many, okay? And so in this example, we're putting, here's one monomer here, and here's the second one. Try to differentiate these by, since I don't have different colors right now. I'll do it by polka dots, okay? So you have monomer 1 and monomer 2, and we're going to synthesize them together by giving off an H on one and an OH on the other. And that's how you get your water, right? So you're removing H and OH to form water, and now you have covalently put together your two monomers, which we're not showing. We're just, remember this R just, it represents like something basically, some odd amount of carbons and functional groups that we don't need to worry about the specifics. The point is that we've linked two monomers together by removing water. We didn't remove water from one molecule, we removed an OH from one and an H from the other, which ends up forming water in the end. Okay? All, and let me repeat, all of the biological molecules in the body, sugars, proteins, lipids, all of these are put together through dehydration synthesis. So those would be the macromolecules that I just mentioned. They are all polymers consisting of many small repeating molecules. The smaller molecules, as we already mentioned, are the monomers. And they are put together through dehydration synthesis. So make sure you write that. All the macromolecules are polymers. They consist of repeating units called monomers, and those monomers are put together by dehydration synthesis, aka condensation reaction. Okay, cell structures and energy sources. So we need macromolecules such as carbohydrates so that we can have energy and we can use their parts to build stuff. Okay, you'll see lots of different types of carbohydrates throughout the cell, maybe in the cell wall, the cell membrane, the organelles. Of course, if we're talking about bacteria, they don't have organelles, but they do have a cell wall. If we're talking about animals, they do not have cell walls, but they have organelles. And other eukaryotes can have everything, right? So carbohydrates will always have this formula of CH2O to the nth time, right? So you can repeat this, it could be a simple sugar that's exactly CH2O, or it could be, if you put a 2 here, then it would be C2H4O2. If you were to put a 6 there, right, then you would have C6H12O6. So you always have this ratio of 1 to 2 to 1, okay? So again, the ratio of 1 carbon to every 2 hydrogen to every 1 oxygen. Okay, so what are the carbohydrate monomers? We call them monosaccharides. They are the simplest of the sugars, right? And they can be anywhere from 3 carbons to 7 carbons. So you can make this in, it could be a 3, it could be a 4, all the way up to 7, okay? Now, you know of deoxyribose from DNA, and ribose sugar is RNA, okay? You may have not have heard of mannose before, but when we work with bacteria, that comes up. Glucose, of course, you're familiar with, because we operate off of glucose and we check our glucose levels in our blood, right, to make sure that we're not diabetic. And if we are diabetic, we are very, very closely monitoring our glucose levels all the time. Fructose, you've probably heard of high fructose sugar in processed foods. Fructose is naturally made by fruits, hence fru for fruit sugar. So, eating fruits are good, but you might want to think twice about fruit juice, which would just be pure sugar. fructose without the fiber. So eating the whole fruit, you're getting all the nutrients and the fiber, and that's good. But just juicing these fruits, then you're mostly just getting the fructose, which is just sugar, right? Okay. Galactose is another type of monomer, monosaccharide to be specific, and we can arrange these monosaccharides in different patterns to get different types of complex carbohydrates. So putting two of those monosaccharides together gives you a disaccharide, right? Di for two. And of course, they're going to be put together by dehydration synthesis, because I already told you that that's always the case. So if you have sucrose, table sugar, the stuff you buy is white, you bake with it, you maybe put it in your coffee, just regular table sugar. Just if you go to the grocery store and you buy a bag of sugar, it's sucrose. So what is sucrose? It's putting glucose and fructose together over and over and over and over again. Okay, what is lactose? You've probably heard of people being lactose intolerant. Lactose is a type of sugar that's found in milk, milk of a cow, right? Not our non-dairy milks. We call things milk that really aren't. Almond milk, soy milk, coconut milk, right? All these non-dairy milks, they don't have lactose. Furthermore, you can get milk from a cow where the lactose has been removed, and then it would be lactose-free milk, right? Anyways, what is lactose? Lactose is glucose and galactose. Some people are lactose intolerant because they don't have the enzyme. The enzyme would be lactase. Enzymes always end with AES. Again, I apologize for this being so sloppy, but I'm just using a mouse. So lactase enzyme is required to be able to break down lactose, to break that bond between glucose and galactose. Okay. Some people make a little bit of it, so they're only slightly lactose intolerant. Others don't make any, and they're completely lactose intolerant. Another sugar you may or may not know of is maltose, and that is when you start to break down a starch, you can end up with maltose. You and I, we cannot break down the starch called cellulose, which is plant starch, but there are other types of starches. And the same goes for some bacteria. Some can and some cannot. So maltose is another sugar that we could test to see what we're working with. What is maltose? It is a glucose plus another glucose. So two glucoses. Disaccharides, like I said, can be broken down by hydrolysis. So everything's put together through dehydration. So everything can be broken back apart through hydrolysis, adding back water. So it's just the reverse of how you put it together. Next, looking at our polymers now. So we looked at individual monomers called monosaccharides. We looked at putting two of those together to make a disaccharide, which is still a tiny molecule. It's still sugar. And then now we're looking at the actual polymers. So we have our polysaccharides, long chains of monosaccharides, joined together, of course, through dehydration synthesis. So examples are starch, like when you eat a piece of bread or a cracker. Glycogen is the way that the body stores up sugar. Wow, I think I need a mouse pad. This is going haywire on me over here. Anyways, glycogen, this molecule here, okay, this is another example of glycogen too, but this is a better example right here. So this example here is just lots and lots and lots of branches And each little dot on the branch is an individual glucose molecule. So you link together glucose over and over and over again to make glycogen, and you can store it in the body. If you try to just keep all of the glucose molecules like separate, individual, then you would end up having lots of water surrounding each of those molecules of glucose, and you would gain so much water weight and you wouldn't be able to move around. So you wouldn't be able to store up very much glucose. But by linking it together into these chains and making glycogen, now you'll have much less water trying to associate or come into the cell. And so that's how we're able to store up lots of energy. You do not store up excess protein, but everything else, fats and sugars, any other excess energy that you have will eventually make its way into glycogen or fat. So sometimes you store up glycogen, sometimes you store up actual fat. We'll look, actually, I don't think we'll actually be looking at that. You'll see that in physiology though. Okay, other types of carbohydrates. I mentioned cellulose, which is a plant starch. It's in their cell walls, and we do not have the enzymes to break it down, so it's fiber in our diet. But these are all types of polysaccharides. Dextrin, we don't really see that too much, but it's another example. Okay, chitin is another example that we primarily only see in fungal cell walls. Sometimes you might see it in another organism's cell wall, but we pretty much, when you hear chitin, you should think of fungal cell wall. It's made up of a very specific molecule called acetylglucosamine. Okay, acetylglucosamine is a polymer. It's a derivative of glucose. Okay, so it's not exactly glucose. It's been modified to be this compound here called acetylglucosamine, and you get lots and lots of that repeating units, and you get this chitin. And there's two types of chitin, kind of like a loose wave and a tight wave, and these will make up chitin cell wall of fungal cell walls. Okay, so also arthropods, which we will mention a little bit in this class because they can be vectors of disease, and that they have very hard shells, exoskeletons. So that property of that really hard exoskeleton is due to chitin. Okay, so typically polysaccharides don't taste sweet, right? When you just bite into a piece of bread, they lack the sweet taste of sugar because all the sugars are bonded together. But if you let it stay in your mouth, like you take a saltine and you just let it sit in your mouth and become very, very soggy, as the amylase enzyme in your saliva breaks apart into the chains into individual sugars, then you'll start to taste the sweetness. So that's kind of cool. You guys could try that later on. All right, we're about halfway through this lecture. I'm going to go ahead and make this part 1, and then we will continue the macromolecules. We're actually much more than halfway done. Anyways, go ahead and take a moment, take a break, and then watch part 2.